Cool. So you'll note that it says three figures above. Where is it? Yeah, three figures. So I actually only described two of the figures in the analyses. So see number 2 where it says create a graph showing the mean and standard deviation, water depth, and flow velocity for each site. That's going to be one graph. The other graph is showing percent EPT and the Hilsenhoff family-based index of biotic integrity for each site. So those will each be two graphs. So one will be water depth and flow velocity. The other one is going to be percent EPT and, as it's called, the FBIBI. All right. So, cool. So we want to compare our two sites here. So Sweeten Creek and Beetree Creek. And the first graph that you'll make is actually going to be with comparing depth and flow velocity. So we'll have depth and flow velocity on the Y. So you might want to think of these as like a two-paneled graph. So like A might be depth and B might be flow velocity. And then you'll have like the mean and standard deviation for each of those for the sites, right? So like, so I think we got like, I can't remember, I think it was like 26 points maybe for Sweeten Creek and like 35 or something like that for Beetree Creek. And so thanks to everyone who entered the data, by the way. So how many of y'all have calculated mean and standard deviation before? Several folks. Okay, cool. So there's a couple ways you can do it. You can do it in either Excel or Google Sheets, but the idea is that like these are what we call descriptive statistics, right? So you want to present what's called a measure of central tendency. In other words, something about the middle of your data. And so one of those we can use as a mean. Have you heard of some other measures of central tendency that are used sometimes? Median, mode, good, yeah. So you could use any of those. And we like to use the mean just because it shows the value that your data, it shows the value from which your data have the minimum error, like the minimum distance from, right? So, median is the middle of your distribution. Mode, of course, is the most common. But the mean is the point from which the total error of your data points is the least. So it's a pretty good descriptor of middle in most cases. I mean, sometimes you'll see medians presented, but not a whole heck of a lot. But whenever we present a measure of central tendency, as it's called, we also need to present a measure of dispersion. So in other words, you're showing like the middle of your distribution and then like how far it's spread out from that distribution, right? Because we can imagine if we had two sites and they both had a mean of like 50 centimeters, and maybe one, like every single point was 50 centimeters, and the other one, like half of them were 10 and half of them were 90, like the mean would be the same, but those would be very different sites as far as the kind of habitat available, right? So we always want to present some measure of dispersion. And standard deviation is a common one with mean because, again, it's showing the gap that encloses about 95% of your data, depending on how you do it. So you guys benefit from me like having put this all on the board before, and I decided to leave it there. But so there are different kind of graphs we could use to present that. And ever think of another kind of graph you might like to use to present, if we put, for instance, water depth on our y-axis, Is there any other kind of graph that you'd like to use besides the bar graph? Anything else come to mind? Or I think a bar graph's probably good. Just trying to think. Does our order on the x-axis here mean anything? Does it matter that we have Sweeten Creek before Beech Creek? It really doesn't. So these on the bottom are what we call categorical variables, right? It just means they're just, it's a category. It doesn't have any kind of ranking. We could put Beech Creek before Sweeten Creek and it would be the same. Now, on the other hand, if we'd quantified, say, percentage of impervious landscape, impervious surface in each of these, and Beech Creek was, I don't know, 6%, and Sweeten Creek was like 28%, then we'd have to sort of standardize the difference between these. But for this presentation, all we're looking at is like difference between the sites. These are just categories we've lumped them into. After we've done that, like, what does it show us? So for your graph, so when you're presenting sort of middles of distribution among two levels of a categorical variable, a bar graph works pretty well. We don't have any lines connecting them, right? We're not interested in the slope between these or anything like that. Just kind of comparing two sites. Can you guys see this through those like plexiglass things? Kind of, sort of. Okay. All right. So you're going to do one of these graphs for depth, one for flow velocity. And I kind of call that one graph. So you'll compare the habitat. You could like have A, depth, B, flow velocity. Then you're going to do other ones for percent EPT and FBIBI. So this is the percentage, and we'll go through each of these, but this is the percentage of, of all our invertebrates that are mayflies, stoneflies, caddisflies. This is an index of biotic integrity based on family. So a family-based index of biotic integrity. And so again, like we'll just be comparing the means between these two sites for each of those variables too. So you'll essentially make 4 of these bar graphs. But if it were me, I mean, you could do them as four graphs. I might do them as like A, depth, B, flow velocity, and call it something like habitat, you know, because that's really why we're interested in studying this, so that it can tell us something about the invertebrates and the fish. And then there's one other graph that I ask you to make for the habitat, and that uses our substrate data. And so our substrate data, you know, we've got 100% of our points. And we want to look at which type of substrate is most common at each of the sites. Why might we want to know about what type of substrate is common at each site? Any thoughts? Mm-hmm. Yeah, so we could look at this substrate and it might help us explain presence of grazing invertebrates or certain fish species. So this is all getting at habitat. So that when we look at the fish and invertebrates and we see differences, we start to wonder why. We can look back at this and see if there are any differences here that make sense. Have y'all made pie graphs before? Pie charts? So for me, I find it easiest to make graphs in Microsoft PowerPoint. Like, I really don't like to reinvent the wheel. So usually what I do is I'll be in PowerPoint, I'll go to Insert Graph, and then it gives me a whole bunch of different graphs to choose from. And I pick the one that looks kind of like what I want to use, and I hit Insert, and then it's linked to a little spreadsheet. And so then you can just go in and change the labels to your labels, change the data to your data. And I mean, I find it easiest in PowerPoint. And I'm not saying you have to, but I think you all get free access to Office like as students, right? So, you know, if you know how to do this in Google Sheets or something else that works better for you, that's great. I personally find PowerPoint pretty easy. So whatever you want to do. But yeah, and then you'll make one pie chart for each site that'll show the composition of each of the different substrate categories. So you'll need to calculate like what percentage of the sites were silt, what percentage of the points at the site were silt versus sand, gravel, etc. That kind of thing. Does that make sense? Okay, cool. And I think we'll have a little bit of time, if you want to, at the end to practice with this. So these are the graphs, and and I put in here too. I don't know if you wanted to, you could do like a stacked graph or something. Just some graph that gets at like percent composition. So like you can glance at it and be like, oh yeah, that site had more silt than the other ones. You know, maybe that's why we saw sunfish or something. Who knows? So yeah, so this is kind of like the habitat piece: flow velocity, depth, and substrate composition. All right. Any questions on that before I go into how we can calculate these two? And just as a reminder, whenever we put a figure in our paper, there should be a figure caption there. We don't need a title on our figure. So you can let that scientific paper pointers handout be your guide, and also just papers that we've read. Like it's all kind of figure and then like caption below it, you know? So I think Microsoft's default, I think, is to give you like a huge title, which like you don't need. Just put in your caption like figure 1 dot mean and standard deviation water flow velocity at Sweeten Creek and Beetree Creek sites. Yeah. Right. Yeah. So I suppose really doing 6 graphs, but yeah. So you'll have one of these for flow, comparing the mean for the two sites, one of these for depth, comparing the two sites, and then the same thing for percent EPT, comparing the two, FBIBI, comparing the two, and then you'll also make, 2 pie charts, one for each site with the substrate. Yeah, good question. Other questions or thoughts? Cool. All right, so why don't I go ahead and open up our Google Sheet, and I'll show you how to calculate some of these metrics. Cool. All right, so here is let's wait on that. So here are our data, and thanks again to everyone for entering that. Oh, you see it a lot better with the projector on, can't you? While we're doing that, I'll just mention, so I'd like you to cite at least 10 papers in your paper, but I've put 5 papers on Moodle. So you can cite those. So that'll be several of them right there that you'll be able to relate to it. And then also any of the papers that we've discussed. Like if you feel like some of those are relevant to your paper, feel free to include those as well. So you may not need to go out and actually find many papers at all if you're, if you're finding what you need. All right, now let's see if I can do this without crashing and burning. So here are our data, and you can see we've organized them in columns with the order and family of every group. So what does the EPH stand for before heptogeneity? Ephemeroptera. Yeah, so this is Ephemeroptera ephemerality, Ephemeroptera heptogeneity, Ephemeroptera cenidae. Then what does the DIP stand for? Diptera, good. And then just moving across here, COL, Coleoptera. PLE is Plecoptera, good, the stoneflies. And then as we keep on going over here, actually I think we're back to the beginning. TRI refers to which order? Trichoptera. So every column heading has an order and a family associated with it, right? Now, sometimes we can't identify everything in the family. I don't know. Maybe it's missing some legs, an appendage, a head, whatever. We might not be able to identify it to family, but we know its order. So you'll notice a couple places here. We have, for instance, this only went to order, Coleoptera. Little redundancy, which is okay. This only went to order Ephemeroptera. So we have some data that we only went to order for, others that we got down to family for. So how do you think you go about calculating the percent EPT for a sample? Exactly, yep. You'll take the total number of Ephemeroptera, Plecoptera, Trichoptera, divide that by the total number of invertebrates for the whole sample, and then multiply by 100, right? Okay. Now, we're going to need to do that for each sample to be able to take a mean for the site. So remember, we had 6 samples per site, right? So you're going to have to do that for each sample, and then we'll end up with 6 values for each site. And then we can take the mean and standard deviation of that, yeah? And if you look at our samples here, you'll notice, what do you notice about these? Several of these have 2 rows per sample. So we'll want to combine those before we start doing our calculations, right? So this is, think of this data, of this sheet as kind of like our raw data. And then if it were me, you know, I go ahead and make another tab with the data that you're working with. In fact, key point here, since it's a Google sheet that we're all working on, the very first thing I do is make a copy for myself and save it like to my own Google Drive, right? To make sure that, have you ever been on a Google Doc or something when someone else is working on it too and you're editing and like, it's like shifting and stuff? So you want your own copy of this that no one else is gonna mess with, right? So be sure and save a copy of this whole sheet to your own Google Drive. And that's what you'll work from. And then again, if it were me, I'd maybe keep this sheet as like our raw data and then make another sheet. And that's gonna be like, you know, maybe like 1 row for each sample. And then like I'll kind of roll with that spreadsheet. So do y'all know how to use like sum commands and stuff like that to sum up? Okay. So I will go ahead and, for instance, I'd come in here and for Sweeten Creek number 4, I'd probably do this. Equals sum up these 2, right? Close the parentheses. So y'all know we type in equals. I'm never quite sure where people are with familiarity with formula in Excel and that kind of thing. So equals tells Excel we're going to hit it with a formula. Then sum, open parentheses, beginning, end parentheses, hit return. And then the beautiful thing is, right at that point, we can just drag this corner and go all the way across. And that'll sum up these 2. And then I would use that for my actual Sweeten Creek number 4 sample, right? Yeah, so well, good question. So we're going to weight all 6 samples equally. So eventually we'll have a percent EPT. We'll have 6 different values of percent EPT, right? And then, yeah, then we'll just take an average of those for our graph. We'll take the mean and standard deviation of those, and we'll all just, yeah, it'll be an average without any kind of weighting. Yeah. Yep. All right. So, so probably everyone understands then how to do percent EPT, right? So if it were me, I'd probably just sum up all the columns that were EPT and then have another column that sums up everything and then kind of roll with it that way. Does anybody want me to go through that, or does that sound like something you might be able to do? Is anyone who'd like me to kind of go through how I might do that? All right, so the easiest way, and again, you guys might have more efficient ways of doing this than I do, but I might do something like this. You know, I'll delete this, but if this were my spreadsheet, I might say, I might have one column which is total, right? And so for that, what I would do is I just hit equals sum, and I think there may be another way to do this. I hit equals sum, open parentheses, highlight the whole thing, right? You see where I am up over here now? And then I close my parentheses, hit enter. There's my total, right? And then I'd grab that corner and drag it down. And there's my total of all invertebrates. Now, remember, I haven't gone back and combined the 2 Sweeten Creek number 6s, the 2 Sweeten Creek. I haven't done that, but that'd be the first step, right, is to combine them by sample. So that's my total. And I don't know, when I have a formula that I've calculated that I think is going to be important, I'm going to keep using it. I might do something different. Like, I don't know, I might come in and I might make this like a little different color, just so I know this is like a little different thing. Okay. Now I want the total number of EPTs. All right. Now this is going to take a little bit longer. So we hit equals parentheses. Here's what I would do. I would highlight this plus, and then start highlighting these. It went all the way across, didn't it? That plus I'm looking for those EPT column. Where's my next EPT column? There it is, right? Scenity. All right. Highlight those. I think those are all my EPT columns. Okay, I'll hit enter. It doesn't like it because I combined it. So, you know what it might need me to do? It might need me to add them one by one. So I do this plus this plus this plus this plus. Tell me if I'm missing any, okay? This plus this plus this plus this plus. Isn't it cool all the different colors that Excel uses? This plus this plus this plus this. And parentheses, bam. Okay. Now, the brilliant magic of Excel and Google Sheets is that I don't have to do that again, right? Now I can check it. You know how I can check to make sure I've got the correct columns selected? Just click on this, all right? Let's look back through here. Go away. Do I have all of my EPT columns highlighted? Let me know if you see any I missed. I feel like I've got them all. We're good. Okay. Then we grab this corner, drop her down. I think there's another pretty color. I don't know. What color should I use, Michael? All right, green. All right. So now I might call this percent EPT. So what are we doing now? What are we doing between these two columns now to get percent EPT? Exactly, right? So equals this divided by that. Let's see if it'll let us do it all at once. We might have to do this in two columns. I don't know. No. All right. Drag her down. And there's our percent EPT right there. A lot of variation, right? We've got several zeros for Sweeten Creek. You know, and we'd need to combine these again. But look, my gosh, at least one of them at Beetree Creek is 100. We've got some 80s, a 90, 73. All right, any questions on how we go about doing that? Does that kind of make sense? All right, so I'm sorry, I have to delete all these. Just the colors to remind you, sort of a Pan-African theme. So we're going to end up with 6 values of EPT for Beetree Creek, 6 for Sweeten Creek. Then we'll take the mean of those and the standard deviation, and we'll use those to make our graphs, right? All right. So the other thing we're going to calculate is the family-based index of biotic integrity. So I'm going to switch out my share here. Actually, no. I want to keep the same one. Okay, so this is one of the papers you'll cite for sure. So this was an article by William Hilsonoff. So different families within an order have different tolerance values, right? So we can go to a little finer level to get a little more data on like the quality of these sites. So there's a lot in here. You can read this whole paper for sure, but if it were me, I'd focus on just a couple things. One of them is his table 1, which takes the range of index values and relates it to water quality. The other important thing is right at the end where he has an appendix here. And this appendix gives tolerance values for different families of invertebrates. And the way this works is invertebrates with lower scores are less tolerant. That makes sense. So the lower the score, the more intolerant your invertebrates are. So what does it range from? Do you see the smallest and large? What are the smallest and largest numbers you see there? Yeah, there's a couple of zeros, right? There's a couple of stoneflies that are zeros. Helgramites, if you guys have ever seen Helgramites, they're a 0, super intolerant, all the way up to a 10, which is like, dude, like they might be living in like the sewage treatment plant kind of thing. And so those are things like a couple types of fly larvae, Psychodidae. Any other 10s? That might be the only one. Oh, you're right. Yeah. Yep. Another diptera larva. So there's a couple fly larvae that are in that category. And so, yeah, so 0 to 10. Now, the first thing I'll say is that you'll probably want a separate spreadsheet. You'll probably want to make a copy of the separate spreadsheet for this one, because we're only going to be able to use invertebrates that we have scores for. So like on this spreadsheet, like delete all the oligochaetes and like the nematodes, and we might have some other families. Whatever we have on there that doesn't have numbers here, we won't be able to use them for the family-based index of biotic integrity. Does that make sense? So again, I'd copy that tab, make a copy of that tab, call it FBIBI, and delete everything that isn't listed here, basically. And then the way this index works is it's a weighted average. So essentially what it's doing is it's saying, what is the average tolerance value of all the invertebrates that you collected? If that makes sense. So what we'll do is we'll multiply the number of each family times its tolerance value. And we'll do that for all the families we have. Then we'll add up that product and divide by the total number of invertebrates to get an overall average. And it basically says like what's the average tolerance score of all the invertebrates at your site? So let me show you kind of how that'll work. Any questions so far? By the way, you guys should put on your resume, like, familiar with calculation of biotic integrity index formulas. Yeah, okay. So I did this earlier with the earlier group, but I'll show you how to do it. So I just grabbed one sample to show you how it worked. I grabbed our Beachy Creek number 3 sample. And then I copied our very first row across, so we have our headers, right? So we've got abundance of all these different families. Okay, so I looked up Trichoptera hydropsychidae. What's the tolerance value for Trichoptera hydropsychidae? 4, right? So these are all the trichopterans here. Here's hydropsychidae, it's a 4, right? So we come over here, put the tolerance value of 4. Now, could cheat and look, you know, let's do this. I need to do one more. Okay, so I didn't do all of them, but just to kind of give you an idea about how this works. Okay. So, what about Trichoptera phylopotamidae? Three. Okay. And now I've set up my formula here to multiply the number times the tolerance value. Okay, so right down here, I've just told it to multiply this times this, right? Okay. What about Ephemeroptera trichorithidae? 4. What about ephemeroptera ephemerality? 1. Has everybody seen that? Ephemerality is pretty intolerant, which is interesting because I feel like we saw a couple of those at Sweeten Creek. I think. I could be wrong, but I have that in my head for some reason. Anyway. Okay, and then I went ahead and did one more. So Plecoptera perlity. What's the value for Plecoptera perlity? It's a 1, right? Good. Okay. Awesome. Okay. So I would set it up like this, right? I just create another row with a tolerance value for each of them, and then multiply. And so this is, again, the number times the tolerance value. Okay, then to take a weighted average, what we do is we sum all of the individuals times their tolerance values, right? So that gives us, whatever it is, 73. Then we sum up the total number of invertebrates. So this number right here, which is 33. And then we divide the sum of the number and the tolerance value by the total number of invertebrates to get essentially an average tolerance value for all the invertebrates. Does that make sense? Cool. So the last step, so after we've summed up the number times the tolerance value, right? So we created a row here that's the number times the tolerance value. After we sum that up for all of them, and I didn't, just for example's sake, I didn't do all of them. There's a couple more down here, but, or no, there aren't. Or yes, oh, sorry, there are. So once you've summed that up, then the next step is to divide that by the total number of invertebrates. So it's essentially giving you like the average tolerance value per invertebrate, if that makes sense. It's like a weighted average, right? So when you do that, it gives us, yeah, and so then we just do this divided by this. And it gives us 2.21212122121. And that's not the real number because there are a couple things on the end there. Yeah, James. So again, yeah, you're going to do this for each sample. And if it were me, I hate reinventing the wheel, I'd copy and paste. In fact, here's what I would do. Let me show what I would do. Okay, so this is BC number 3, right? If it were me, I'd copy and paste this whole dealio down here. Okay? Then, when I'm going to do BC number 4, all I got to do is grab our numbers for BC number 4, right? And we don't have too many for whatever reason. That's a good point. So let's just pretend that this is all of BC number 4. So I'd highlight the total number of invertebrates, and then I'd do just Control C, right, to copy it. Jump back over here, right here, right, because this is the number, right? This is my number across here. Hit Control V. And as they say, voila. Exactly. So we're only going to end up with 6 for each one, because our samples are standardized by the server sampler. So we only really had 6 standardized samples for each. But in the lab, for identification purposes, sometimes we split those up a little bit. So yeah, so you'll sum them up. So yeah, I would go in here, and did we show you kind of how I would do it? I would just come in here and I'd say, I do this. I would say, for BC number 4, I do this. First of all, I'd do something silly like that to get rid of that. I do this. I go insert 1 row above. And I would do this. I'd say equals sum of what? These 2, right? Because 6 and 7 are my BC number 4s. Does that make sense what I'm doing? Yeah, and then I just hit enter. Then I would grab the corner and drag that thing all the way across. Then I'd call that my BC number 4, and I'd delete these 2. But before I do that, what would I do? See if anybody can catch me. Save, but notice that this is just a formula based on these 2 cells. So at this point, if I delete these 2 rows, what's going to happen to this row? It's going to, well, it probably just give me zeros, right? So the one next thing that I want to do is I'd want to do this. Right click, copy, right click, paste special. Because you all are special, and so am I. And what am I going to select? Values only. Okay? Now, it's still got the values, but look up in the f of x bar. Right? It's got, where are we? How can I see that? I'm blanking out here. Sorry, yeah, right. It will have, yeah, sorry, it has 4. Whereas if we look up here, what's it got? Some C6, C7. So as you're going through and you're creating new rows or new cells from formulas, before you delete the cells you used to make those formulas, you gotta copy and paste your new cells as values only. Because otherwise that's just a placeholder to calculate, right, based on other stuff. So, yeah, I've screwed that up before. Many times. It's not fun. All right. So I'm going to delete this because we were just playing around. Yeah, so again, if it were me, let's keep this as our raw data. I would copy this spreadsheet into this Google Sheet into your Google Drive, start playing around with it, and then go from there. Yeah, you guys are all going to be like spreadsheet jockeys before we're done. Any questions about what we're doing there? So again, if it were me, right, I would keep my formulas and everything here, just copy and paste them. Only thing I'm going to do is paste those new numbers in there and let it calculate it for me. Now, if I copy a new, and you know what, again, just to keep myself straight, I would probably make these a different color just to realize what I'm doing, right? So these are my actual samples. I'd probably do something like this. So now if I copy and it has some families in here that I don't have tolerance values for, I'll have to do those again, or I'll have to do those. But if it's got things like hydrocyclids and phylopotamids, which I've already got tolerance values for, it's going to calculate that for me automatically. In fact, it gave me my FBIBI for BC number 4, and that's the only thing that was in this sample. And just for grins and giggles, let's look and see what those values mean according to Hilsenhoff. So a score of 2. What is a score of 2? I had it highlighted from earlier. So a score between 0 and 3.75, Hilsenhoff says, is excellent. On the other hand, that 4 value that we got is very good. So anyhow, it's on the Moodle page. So I created a separate, in fact, let me go back here. So so I grayed out some of these sources because they're for the other paper, but so that's this Hilsenhoff paper right here. This Weigel et al. Article uses %EPT, so you might find that useful to cite. This paper looks at differences in invertebrates based on land use, so you might find that interesting. This Jeremy Tiemann, oh shoot, I forgot to mention. So Jeremy Tiemann, who I did my master's with, is coming. He's going to give, no, he's not coming here, but he's going to be giving a departmental seminar on Thursday. So you guys should check it out. It's pretty cool. And you can email me with a summary for 10 points extra credit. But anyway, this is the paper he wrote, not in the year 204, but in the year 2004. And it uses %EPT as well. And here's the final one I'll talk about, is this one right here. So any questions so far on how this goes? Yeah, I would just go here, Torrin, like I would just go file, make a copy, you know, and that'll copy the whole spreadsheet and all the tabs. And that way, yeah, because y'all are going to be doing different calculations and things. Sometimes it's great because you're like, oh, that makes sense the way they did it. Other times it's like, that doesn't make any sense at all. Cool. All right, so this is just like my little kind of test. So does everybody understand kind of what's going on here, how we go about doing that? The way I learn things is just playing around with them, just playing around with numbers to see what it does. Like that's really the way to learn, you know? So if you're having any trouble, I've got office hours tomorrow, I've got office hours Thursday, my schedule's pretty flexible Friday, I've got office hours Monday, so feel free to drop by with this. And I think if we do Google Hangouts or Zoom, I think we can like share a screen and you could like show me what you're doing and stuff if you run into trouble. So so feel free to do that. Okay, and then the last thing that I wanted to look at. What? Yeah. Yeah, so we didn't get any of the blood red chironomides. The ones are red, they're blood red, which makes them better at acquiring oxygen, which they need to live in low oxygen environments. But we didn't get any of those, I don't think. I think all the chironomidi we got were the other ones. Cool. All right, so last thing to talk about, for the fish, no calculations, no graphs. I'd just like you to make a table that shows common name and scientific name and abundance for the fish that we got, for the two of them, for the two sites. And then I'd just like you to take a look at this source. So this is a paper written by some folks that work at the TVA, Tennessee Valley Association. And they used it to calculate an index of biotic integrity. This is, see in here where it says the Saylor and Scott 1987 paper. This is on Moodle as well. A lot of stuff in this paper, but really all you need to worry about are their characterization of these species as I intolerant or T tolerant. And so this table has a lot going on. There's a lot of stuff here. And there's a lot of species here that we didn't get. But there's something we did get. So like stone roller, big-eye chub. We might have gotten a whitetail shiner or two. So just look through this list, and they don't say tolerant, intolerant for all of them. So either T or I is tolerant versus intolerant, okay? So for instance, let's see, they say, find one that we have. They say that green sunfish are tolerant, right? They say that and some other ones that we have. Creek chub is tolerant. So there's actually not a whole lot of ones that we got that they listed here. But basically just look over the characterizations of them for fish that we got, which are not going to be too many, to be honest. And just see if you see any big differences between the sites, which, to be honest, I'm guessing you won't, because they have categories for so few of the species that we got. But just kind of take a look through it. And they have other information here on what they feed on. So IN are insectivores, O is omnivores. You know, so they've got other stuff here, which I don't know. Maybe for your second draft, you could go into more detail on that if you wanted to. But for this initial draft, looking at tolerant versus intolerant fish species and seeing if you see any difference among the two sites, which might or might not. Just kind of an eyeball qualitative kind of thing. Don't need any graphs or calculations on that. But I would like you to make just a table of the fish species, basically. So cool. Questions, thoughts, comments thus far? Yeah, James. Yeah. So if there are families that we got that aren't in there, yes, then also we'll have to omit them. Yeah. And like I said, I haven't gone through them all. So yeah, with the FBIBI on that Hilsonoff paper, yeah. Just completely exclude it. Yeah, completely exclude it. Because to calculate the FBIBI, like you have to have, like you have to have a value. So, right. Oh, okay, yeah, nice. Oh. Yeah, I guess that's true. Yeah, I guess that's true. You wouldn't, I guess you wouldn't have to, I mean, you're right. So I guess that's like my, more like anal retentiveness. Like I'd delete everyone that I wasn't calculating for. But if you want to leave them in there, realizing that, what you say is true. Yeah, I guess that's true. Because so you're right. So it wouldn't give you like the sum of the abundance times the FBIBI, but it would still sum up the abundance. You know what I mean? So if you left them in your list, you just want to be sure and delete the abundance too. Yeah. Because otherwise you'd get an artificially low FBIBI because you'd be dividing by a higher number than you used to calculate. Is that so? In other words, if you left them in your spreadsheet, but you couldn't calculate the FBIBI, you'd have 0 for the abundance times the FBIBI, right? Because you didn't have a tolerance. In other words, over here on the so like on the calculations here, like let's just say Peltoperlity, let's just say we had 3, right? But it doesn't have a value here. So because of that, there's no value here from Peltoperlity, right? But it's still going to be counted in my abundance. And so, in fact, see how this value down here changes a bit? So yeah, I think the take-home message is either delete the columns or at least the numbers associated with them. Yeah. Europe. Other thoughts? Oh yeah. So you guys, I'm looking forward to seeing these results. Man, just eyeballing the data. I'm guessing the invertebrate-based metrics are probably going to be pretty different between the two. And the fishes, I don't know. Maybe for your second draft, if you want to look some at like, you know, and if you want to say like, there are more of these fishes at this site than the other one, that's cool, but we don't have tolerance values for a lot of them, so we won't be able to say like what that means about like the quality of it necessarily. There are other papers that have more tolerance values, but I don't think I'm going to throw those at you guys right now. So you'd include those in the percent EPT just because you can do that different ways, James, but normally it's just done with like all the invertebrates you get. The FBIBI, right, yeah. So FBIBI is only insects. Or wait, there may be a crustacean or two, but there's definitely no nematodes. There's definitely no nematodes or like oligochaetes. But yeah, but here's that source, that Hilsonov source. Yeah, so they have amphipods and isopods in there, which are crustaceans, but it's almost exclusively insect families. Yep. Cool. Any other questions? Thoughts? Oh, shoot. I forgot to show you how to do the t-test. Almost forgot, but not completely. Yeah, you can use whatever you want to run it. Absolutely. So, how many of y'all have done a t-test before? All right, so a t-test is a way of testing for a difference in the means of two groups. So if you have more than two groups, anybody know how you test for differences in those means? Yes, analysis of variance, aka ANOVA. So if we had 3 or 4 or 5 or 6 or 7 sites, to test for significant differences between those means, we'd have to use ANOVA, analysis of variance. But since we only had 2, we use a t-test. Okay, so again, if you have a software package that you like to do to run t-tests, I'm totally fine. You don't have to do it this way. This is just kind of a path of least resistance that I've come across in Google Sheets. Now, go to add-on XL Miner Analysis Toolpak. Now, the bummer we found out in the earlier session is that apparently your UNCA Gmail accounts don't let you add this on without like a note from your mother or something. I don't know. So if you have a personal Gmail, this is probably how you want to add that. And if you don't, I don't know, send ITS an email. I can't believe that they don't let us add it, because we can add it onto all the university computers. I don't know why you couldn't do that on your individual account. It's kind of ridiculous. I can call them if y'all really want to use it on your UNC account. Otherwise, you can do it through your personal one. So it's called the XL Miner Analysis Toolpak. It's really easy to use, and I'll show you how it works. So you go ahead and you start that up. And then you see it gives you a bunch of options for different statistics. You just keep walking by all those options until you get down to the t-test options. And you can see there's a few different t-tests. The one that is used almost all the time is this 2-sample assuming equal variances. It's the most straightforward, easiest to use, so you click on that button. So you see it asks for the variable 1 range and the variable 2 range. And so I just made up 6 numbers for Sweeten Creek and Beetree Creek. And these would be like the FBIBI values. Like that's kind of the ballpark that we're in here with these kinds of numbers. So for variable 1 range, the way it works is you highlight them, then you click on it. So there's variable 1 range, A2 to A7. Then we come over, highlight these. It's variable 2 range. And then you don't have to worry about any of this. The alpha just means that we're only going to call it significant if the p-value is less than this number. And so since it's 0.05, that means that it'll tell us it's significant if it's less, if p is less than 0.05. That's the default. You don't really have to worry about that. The main thing is just getting your variable 1 and variable 2 range in there. Then it asks for an output range, like where you want it to put your results, basically. And so just click anywhere around here and click on output range and hit OK. All right. And so you can see it calculates the mean for each of the 2. So the way I set this up, the Sweeten Creek mean was 2.6. Beetree Creek mean was 4.9. The variance was identical. But really, all that you're interested in is this value down here. P of t, it's your p-value for a 2-tailed test. Now, can anybody tell me what a 2-tailed test is? Think of it as like the stonefly test, because it has 2 tails. Right, exactly. So 2-tailed means we're okay if our value is in either tail of the curve of the t-statistic distribution. So we don't care if Sweeten Creek has a higher number than Beetree Creek, or if Beetree Creek is higher than Sweeten Creek. Question. Even on your personal account? Oh no. That's weird. You need the analysis toolpak or something silly. Does anybody have Excel on their machine? Can they look and see real quick? Do you have like the UNCA version or whatever you got from our IT office? This is really annoying. Oh, it doesn't say why, huh? Okay. Oh, it's so frustrating. So you can see we installed it on this computer here. But is it? No, it's my account it's installed on, not this computer, right? Okay, good. All right, so it does, at least the version on this machine has the t-test function. So if are you in Excel curious? So when you type in equals t-test, does it act like it knows what that is? James? Okay, so that's just like it does it in Excel then. Okay, yeah, all right. So all right, well let's try it. Let's try that then. Yeah, so that's the way you do it. You set it up in Excel. All right. Goodbye, Toolpak. So yeah, equals t-test. And then I think it's like the first array, right? So it's this, comma, this, comma, the number of tails. Is that right? Now, yeah, okay. So to be honest, this might be simpler. Well, I don't know if it's simpler. It's less stuff to figure out. But anyway. Yeah, so okay, so we'll just go here step by step. So you type in equals t-test, parentheses, highlight your first set of data, comma, highlight your second type of data. Then the next thing it asks is the number of tails. And so again, as we said, we want 2 on there. Yeah, and then, Michael, when it asks about the type of t-test, that's where we'd have to see how it's classifying it. Okay, so there's 3 types, just like on the data miner. The first is a paired test, which we're not doing. The third is unequal variance, which we're not doing. And so the second is the equal variance assumed, which we do. So yeah, so as far as the syntax goes, then it's the number in this cell is going to be the p-value. It's not going to give us anything except the p-value. Yeah, so equals t-test, parentheses, first group of data, comma, second group of data, comma, 2 because they're 2-tailed, comma, 2 because it's a 2-sample t-test. All right, thanks, James. Yeah, so that, okay. Then when we hit enter, did it give us all this? No, this is there before. Right? This wasn't there before, was it? Or it was. Sorry, you know what I mean. Okay, so the number that it will give you is this. This is the p-value of the null hypothesis. I mean, this is the p-value associated with a hypothesis of no difference between these two. So do we have a significant difference here for the data that I made up? You had a 50-50 shot, Brian. I explained it poorly. So this value here is the p-value associated with our null hypothesis. And the null hypothesis is that those means are the same. So since our p-value here is super duper low, we'd actually conclude that there is a significant difference. My apologies for not explaining that correctly at all. So if this was 0.04, we would say it's significant. But since it's 0.00004, it's 1,000 times significant, if you want to think of it that way, which is kind of weird. So if it were me, I would set my data up just like this. When it's all said and done, and you've got your FBIBI for the 6 samples in Sweeten Creek, the 6 samples at Beachy Creek, you got your percent EPT for the 6 samples at Sweeten Creek, 6 samples at Beachy Creek. I'd set them up just like this and type in your t-test syntax just like that. Now, This doesn't give you the means, right? Like the other way did. And you're going to need the means to make your graph. But, I mean, you know, y'all know how to do that. Probably, all were probably born knowing how to do that. This generation of these young whippersnappers, they just know all this data stuff. A lot younger than I did. Back in my day, if we had to count more than 10, we had to ask for a friend to come over because we ran out of fingers. All right. So I need to send out a little note about that. Or I could just tell them to watch the video. Sorry, does that laugh evil? I didn't mean that to sound evil. I wanted it to sound joyous. Yeah, all right. Any other questions about this? Well, I personally am looking forward to reading your papers. So, yeah, let me know if you have any questions. It'll be awesome.